Hello guys, and welcome to notes number 12. We just finished up mitosis, and now we are moving on to the process of transcription, which involves 3 types of RNA. We're also going to be looking at translation and mutations. So we're going to first be talking about transcription. Now it's important that this is not the same thing as replication. What transcription is it's copying DNA into a strand of RNA. Okay, so this is just going to be a single strand that we're making, and it's a complementary or opposite strand of RNA. And transcription is going to occur one gene at a time, depending on what gene we need. If I need a gene that's going to synthesize more skin cells because I'm lacking those and I need to replace them, transcription is only going to make that gene. And it requires a specific enzyme called RNA polymerase. What RNA polymerase does is it allows the molecule of DNA, which you see here, and this is the enzyme, to open up the strand of DNA and add on a complementary mRNA strand. Okay, so if you remember, RNA is always single-stranded, and it's complementary because, remember, G and C always pair together, and A and T always pair together. Okay, so say this is the gene that you need to make. Okay, again, DNA polymerase, this green enzyme, is going to unwind the DNA. It's going to unwind the DNA and add nucleotides on. Here's another illustration of what transcription is. It's just zooming in to the DNA molecule and showing that RNA is made. And basically the reason, just going back for a second, the reason why we need to make this single strand is that it's too big to leave the nucleus. So since DNA can't leave the nucleus, we have to make a messenger in order for it to squeeze out of the nucleus and go where it needs to go to make proteins. So let's just distinguish between the types of RNA. There are 3 kinds of RNA, and each one is going to play an important role, but they're all involved with protein synthesis or making proteins. They are messenger RNA, which is represented by mRNA, ribosomal RNA, which is represented by rRNA, and transfer RNA, which is tRNA. Messenger RNA is what we just saw. It's taking the message about what protein we need to make from DNA, which is in the nucleus, and it's going to send it to ribosomes in the cytoplasm. Okay, remember, ribosomes are what are making proteins. So we need to make a messenger in order to be able to squeeze through our nucleus. Ribosomal RNA is the RNA that makes up ribosomes. Remember, ribosomes are what make proteins, so these are very, very important in the protein synthesis process. Ribosomes are also made of proteins, but the most important is that it's made up of ribosomal RNA, and this is what a ribosome actually looks like. Usually we just represent it with little dots, if you remember when we were doing the cell. But if you were actually to zoom in on those dots, this is what a ribosome would look like. It kind of looks like a weird tooth. And it is what is going to allow the messenger RNA, right here, the messenger RNA, to make proteins. You can see a protein being made right here. And transfer RNA. Transfer RNA is going to carry an amino acid to the ribosome. So if you remember, amino acids are what make up proteins. Okay, so basically this is what transfer RNA looks like. It has this weird shape, and it carries an amino acid right up top here, depending on these 3 nucleotides right here. So if you remember, the nucleotides are A, T, G, and C. A and T always pair together, and G and C pair together. So if this code, say, is GCA, then it's going to represent a specific amino acid right here. Okay, so remember, if I have an amino acid plus another amino acid plus another amino acid and keep going, this is going to become a protein. So, as I just said, proteins are made up of repeating units called amino acids. So here we have, instead of representing them as circles, we have them with squares, and these are just the short abbreviations for the different types of amino acids. If you recall, there are 20 different kinds of amino acids, and they are going to link together to make a protein. So here's our tRNA represented a slightly different way, but it's also going to be with this little code, taking an amino acid, transferring an amino acid. So the order of amino acids is important for a protein to function properly. So say this is a protein right here. If I switched this amino acid with this amino acid, it would completely change the protein and it wouldn't function properly. And the sequence of amino acids is determined by the messenger RNA, by the genetic code. Okay, so like we saw here, this 3-letter code has to match up with the message that we got from the original DNA. Okay, so each 3 letters is called a codon, which codes for a specific amino acid. So say the mRNA right here says CUA, that means that the anticodon right here, this code that matches up, is always going to bring this amino acid LEU. Okay, and this sequence CGC is always going to bring the amino acid ALA. So it's kind of like a secret message that DNA has with these letters or nucleotides. Each 3 is a codon that's going to say, yes, I need this amino acid, whatever amino acid it is. I'm trying to write amino acid, but it looks retarded. Okay. And basically, we just have to review what a gene is, right? We said that each gene is transcribed individually. And looking at it in the larger picture, this is a chromosome right here. Chromosomes are made up of DNA, very long strands of it, like that string we called chromatin, wound up. And each segment that codes for something, so say the gene for blue eyes or the gene for height, is in a different segment. And these genes are what are going to determine what proteins will be made, right? The genes are the code. They are the ATGC that are going to tell the transfer RNA what amino acids to bring. Okay, so when a cell needs a particular protein, we have to go about this process called protein synthesis. And we're going to kind of sum this all up again one more time. So we're going to review a little bit. Okay, so that first step that we talked about was transcription. So this happens inside the nucleus, because this is where DNA is where it is. Okay, and then the gene that needs to be transcribed into that secret message is going to be transcribed into a piece of messenger RNA. So just like we saw before with the double strand of DNA, that enzyme DNA polymerase is going to come along, open up this strand, and make a message or a copy of it, of mRNA. Then that mRNA, since it's single-stranded, is going to be able to leave the nucleus, and it's now going to go to a ribosome in the cytoplasm. Okay. So once that mRNA is on a ribosome, now we can start decoding. That genetic code that mRNA has is going to start attracting our tRNA molecules, which have those specific amino acids for each 3 letters. Okay, so again, DNA is inside of the nucleus, but it's too big to get the message out to the ribosome. So in transcription, we make a piece of messenger RNA, which goes to the ribosome, which is going to start making a protein. Okay. Now, translation is all about how we decode those three-letter codons, right? We're translating the code from letters or nucleotides to amino acid. Okay. So it's based on that code, like we saw, those three letters, which tRNA is going to drop which amino acids. Okay, so these amino acids are going to attach to each other. They're going to keep going, three letters, each an amino acid connecting to make a protein. Now, all of this is happening in the ribosomes. Remember, ribosomes are where proteins are being made. So this is the actual process of how ribosomes make proteins. So distinguish these two terms. They sound similar, right? We just learned about transcription. Transcription made DNA into a copy of RNA. And now translation is taking this RNA and making it into a protein, and taking the code and I'm decoding it. I'm translating, just like I would translate a language. Okay, and again, one more time. This is DNA. It's just showing one strand to simplify it. Transcription is making messenger RNA, and then each code or codon is then going to signal a certain amino acid, which is going to make protein. So this process is called translation. I'm translating the code. Okay. Now, sometimes we have problems with translation or transcription because mutations happen. Okay, just like you would think of a mutation in X-Men or any sci-fi movie, it's a change that's happening, and it's a change that's happening in the nucleotides. It's changing the sequence. So here you can see that there's ultraviolet light coming in and harming this DNA, and it is destroying the DNA. So sometimes mutations can result in different proteins being made. If something is changed in the DNA level, that's going to change the protein. A point mutation is a mutation in only one single nucleotide. Okay, so if I have a long strand of nucleotides, A, T, C, G, A, G, let's say. Okay, it is just taking one of these letters, say this T, and replacing it with a C. This can change the amino acid. It can change the whole code. But it's not always bad. It can be bad and cause a genetic disorder. But sometimes point mutations or any mutations can be good. For evolutionary sense, it could increase the chances of survival, right? It could change the color of an animal because it changes the protein that's being made, but it could end up being good because they could blend into the environment. So it's not always bad. So there's different types of point mutations. There's one, which I just demonstrated, which is substitution. One nucleotide is replaced by another. So in that long strand, if I had a T, I would just replace it with a C. Okay, so this is what sickle cell disease is caused by a substitution mutation. Insertion is if a new nucleotide is added in. So if I have that long strand, A, T, C, G, A again, okay, if I were to just add a nucleotide, let's just put it in right here and add a T, okay, I'm changing my code, right? Now I have these codons, this is a codon, and so forth. Huntington's disease is caused by an insertion mutation. And last is deletion, which one nucleotide is removed. So instead of adding one in, I would take away, say, the G. And again, that would change how I read my code. So our insertion and deletion, those last two that we just looked at, either putting a letter in or taking one out, are considered frame shift mutations because they changed the code. They change the way the codons are read, and they completely change everything. So here we have an example of DNA being transcribed into mRNA and what the amino acids would be if it was read with these codons. But now we're going to see that a substitution is going to happen here. Okay, so I'm substituting G for T. Okay, so all this did was change one little thing. So this is still the same codon. This just changed a tiny bit right here. And this is the same, and this is the same. So only this one amino acid changed, right? The rest are the same. You still have met, thur, and u, whatever those are. Those are abbreviations for them. So this substitution is not considered a frame shift mutation. It didn't change everything. It just changed one. But if I insert a letter, okay, so here I inserted what's going to be transcribed right here. Okay, I added one. Now my codons are completely different. Instead of having AUG, I have AUA. Instead of having CGU, I have GCG. Everything after this is completely changed. So you can see that my amino acids, which I should have made, are completely different. None of them are the same, besides the last one. Okay, and the same thing with deletion. I demonstrated it in a funny way. Obviously, we're not using these letters, but if I have the saying, the fat cat ate the rat, which are each 3 letters, a codon, and I delete the H, all of a sudden, all of my codons change, because I have to have 3 letters, and all of a sudden I can't even read this sentence. And that is it for notes number